Hello everyone, welcome to the ITLS Academy. Today we are going to perform isolate extraction of DNA from bacterial cells. So these are the chemicals which are required for performing extraction of DNA from bacterial cells. The very first is 10% solution of SDS. SDS is what? Sodium dodecyl sulfate. Phenol, chloroform, lysozyme and buffer solution, distilled water, and with that we also need the solution of sodium acetate. Okay. So first of all, what we are going to do, we need to, the very first step is take 1 ml of distilled water in the epidrauf, and see, you can see we have already marked this in the pipette, and now this is the epidrauf. Now I'm going to pour the water, measured water in this. And we need to fill all the 12 epidrauf with the same amount of distilled water. Here we have already filled each epidrauf with 1 milliliter of distilled water, and as you can see I already mark all the epidrauf with B. See, B is indicating that we are extracting the DNA from bacterial cell. Now with the help of inoculating loop, I'm going to extract the bacteria from our culture and pour it into the epidrauf for making culture broth. So by the same procedure, we need to fill all the 12 epidrauf, and after that we need to centrifuge all these epidrauf at 3000 rpm for 10 minutes. Okay. So as you can see here, our epidrauf is ready with the bacteria. As you can see it here, is it visible to you? See, you can see here, this is a white color bacteria. So we already inoculate the bacteria in the epidrauf. Now by the same procedure, we need to perform all these 12 epidrauf. After filling the bacteria into the epidrauf, now you can see these white pellets clearly. Okay, what we are going to do, we are going to need to centrifuge at it for 10 minutes at 3000 rpm, as you can see. Now place all the epidrauf carefully and make sure that you can balance it properly. Here you can see the mouth of each epidrauf is it in the towards the center. So now here we are ready to go. As you can see the time, it's 10 minutes and 3000 revolution per minute. So now I'm going to start it. After 10 minutes you will see the result. So friends, after performing the centrifugation, now we have take it out from the centrifuge. What we are going to do now, we need to discard the supernatant and we take further pellets. Okay, so for discarding, you remember that pellet should not disturbed while you're discarding the supernatant. So here you can say discard the supernatant, and now only the pellets are remain there. So by the same procedure, we need to discard all the epiandrops, and after that we work with the pellets. Okay. So after discarding all the supernatants, pellets are only remain here. Now very first step is we need to pour this TE buffer, 0.5 ml of TE buffer in each of the epandrop, and for that I set 500 microliter on this. Now with the help of micropipette, take 0.5 ml of TE buffer and add it into each epandrop, and by the same measurement we need to add this buffer solution in each epandrop. So after adding 0.5 ml of TE buffer to each epandrop, now what we are going to do, we need to add 75 microliter of a 10% SDS solution in each epandrop. So after adding 75 microliter of 10% SDS to each epandrop, now we need to add 7.5 microliter of lysozyme to each epandrop. So here we have already set 7.5. Now I'm going to take 7.5 per microliter of lysozyme and pour it into each epandrop. Okay, by the same procedure, we need to add it all the epandrops. So after adding 7.5 microliter of lysozyme in each epandrop, we need to incubate at room temperature for at least 1 hour. Okay. So after incubation at 37 degrees Celsius for 1 hour, now we are going to add 0.5 ml of chloroform and phenol in each epandrop. So I already set it at 500 microliter. Now I'm going to add 0.5 ml of chloroform in each epandrop. So here we go. Now in the same epandrop, we need to add 0.5 ml of phenol. So by the same amount, the same amount we need to add in each epandrop, 0.5 ml of chloroform and 0.5 ml of phenol. So one more thing which I would like to add, that you need to boil the phenol in water bath. So after adding chloroform and phenol to each of the epandrop, now we need to refrigerate these epandrop for 15 minutes, and after refrigeration we further need to go with centrifugation at 5000 rpm for 15 minutes. So after centrifugation we are going to saw the results. So after refrigeration for 15 minutes and after performing the centrifugation, now we need to discard the supernatant of each epandrop, and after discarding the supernatant of each epandrop, we need to add further 0.5 ml of chloroform and 0.5 ml of phenol in each of the epandrop, and further refrigerate it for 15 minutes, and after performing the refrigeration we need to undergo centrifugation at 5000 rpm for 15 minutes. But you need to make sure that while you're dealing with the phenol, you wear proper gloves, mask, and all these things. It would be helpful for your health. So after performing the centrifugation, now I am going to discard the supernatant, and we need to carefully discard the supernatant so that pellet should not damaged. So after discarding the supernatant, now I am going to add 100 microliter of sodium acetate in each of the append drop. So during discarding, we need to make sure that pellets should not be damaged. As I already set on micropipette 100 microliter, now I am going to add this amount in each of the append drop. We need to make sure that we can pour this amount very carefully in each of the append drop, and after that I am going to add 200 microliter of chilled ethanol in each of the append drops. So as you can see, I am pouring sodium acetate in each of the append drop. So after adding 100 microliter of sodium acetate in each of the append drop, now I am going to add 200 microliter of ethanol in each of the append drop. And after adding 200 microliter of chill ethanol in each of the append drop, we need to keep these append drop for few minutes, and after that we need to discard the supernatant. So after adding chill ethanol, now I'm going to discard the supernatant, and after discarding the supernatant, I'm going to add 50 microliter of tea buffer in each of the append drop. So after discarding the supernatant, I am going to add 50 microliter of TE buffer, and after adding this TE buffer, we are going to store the append drop. So after adding 50 microliter of tea buffer in each of the append drop, now our process of DNA extraction is completed. Now we are going to store these in the refrigerator for performing the further processes like PCR, gel electrophoresis, etc. Hope you got it.